Hello everyone, we are Subgroup 1 from Group 2. We would like to present our Assignment 1, Title 1, Laboratory Safety and Incident Response Simulation OSHA. In this title, we are focusing on responding to biological spill. Here are some safety regulations stated from OSHA for biology lab. First, employer must prevent from mouth pipetting and suctioning of blood. Besides, employees should not eat, drink, smoke, apply cosmetic in the work areas. Next is, please wear gloves when dealing with blood, body fluids or contaminated items. Last but not least, the workers exposed to blood must get a hepatitis B vaccine under medical supervision. The personal protective equipment (PPE) standard requires the employers to provide and pay for PPE when there are hazards that could cause injury or impairment. Based on the assessment, the employer must select appropriate PPE such as protection for eyes, face, head, extremities, protective clothing, respiratory protection, shields, and barriers to protect them from hazardous materials. Then, employers must provide training for workers on how to wear and care for it and its limitation also. Furthermore, we should keep a spill kit handy in the laboratory to quickly clean up a biological spill. The spill kit should include the concentrated disinfectant such as chlorine bleach or Lysol, the packages of paper towels, the household rubber glove or utility glove, the forceps to pick up the broken glass, and also biohazard bag to safely collect and dispose of materials contaminated with biological substances. Biosafety Level 1 is applied to labs working with low-risk microbes, such as non-pathogenic strains like E. coli. Research is conducted on batches without special contaminant equipment. For this biosafety level 1, the safety protocols we have to know is we need to use mechanical pipetting. Mouth pipetting is not allowed. Avoid splashes or aerosols. Decontaminate work surfaces daily after work. And lastly, no food, drink or smoking materials are allowed. Emergency response for blood spill. First thing we need to do is wear glove and lab coat, use tongs or mechanical means to pick up broken glass or sharps and then absorb the blood with paper towels, clean the spill site with the detergent solutions, wipe down the site with paper towels, soak in the chlorine bleach solutions, and then discard all the contaminated materials in biohazard waste container. Lastly, we have to wash our hands with soap.
Hello everyone, our video will explain the proper way to clean up spill inside the laboratory. The laboratory that we are using is the Plant and Environmental Biotechnology Lab. In this scenario, our protagonists well trained and prepared, take every step with precision to ensure the safety of themselves and others in the laboratory. Right from the start, our protagonists prioritize safety by wearing the necessary PPE covering eyes, nose and mouth to prevent exposure to the biohazardous material. When encounter a spill, the first thing to do is to keep the spill from spreading by covering it with paper towel. The common type of disinfectant employed in research laboratory is 10% bleach, which in this demo 7% ethanol was used as an alternative. After soaking the spill with ethanol, it is recommended to wait for 20 to 30 minutes before swiping. While waiting for the spill to soak, prepare a biohazard bag to throw away any contaminated material. Then dispose of all contaminated material including paper towel and broken glass. Ensure the biohazard bag is placed inside a dedicated sharp disposal container.
In case of contamination in the lab, the response involves several key steps. Firstly, evacuate the area promptly to ensure everyone's safety. Secondly, isolate the contaminated area by sealing it off to prevent further spread, utilizing barriers or closing door. Next, notify authorities immediately, informing the lab supervisor or relevant personnel about the incident. Then, follow protocols in handling the contamination by adhering to established lab procedures, which may require specialized cleanup and protective equipment. Prioritize personal safety by following decontamination procedures if trained and equipped before leaving the area, preventing the spread of contaminants. Lastly, document the incident comprehensively, detailing what occurred, the involved substances, and actions taken, as this information is vital for investigations and enhancing safety measures.